so glad you are here. My name is Laura Douglas, and I am the Grant Program Manager here at the Arthritis National Research Foundation. On behalf of our entire team, welcome to See Beyond Dryness, Understanding Sjögren's as a Whole Body Condition. This is the second webinar in ANRF's Arthritis and Lupus Awareness Month series, and we have a truly exceptional program ahead of you today. Whether you're joining us from rheumatology, ophthalmology, primary care, or another specialty entirely, this session was built for you. I want to take a moment to thank our supporter, Johnson & Johnson Innovative Medicine, for making today's program possible. It's because of support like this that we are able to bring high-quality, independent education directly to healthcare professionals. Thank you. A bit of housekeeping before we begin. This webinar is being recorded and will be available on demand. We'll share the link in a follow-up email, so please feel free to pass it along to colleagues who couldn't join us today. This live session is CME accredited. Please stay tuned to the end of the program for instructions on how to claim your credit. If you have any questions for our faculty, and I hope you will, please type them into the Q&A box at the bottom of your screen. We'll have dedicated Q&A time throughout the hour, and I'll be reading your questions directly to the faculty. If we run out of time, please email us at info@curearthritis.org, and we'll do our best to follow up. We'll also be running a few short polls throughout the session. They help us understand your experience and shape the conversation, so please do take a moment to respond when you see them. Now, a little bit about today's program. Sjögren's disease is one of the most common autoimmune conditions and one of the most commonly misunderstood. For too many patients, the conversation stops at dry eyes and dry mouth. But the reality is far more complex, and the systemic manifestations of the disease help shape patient outcomes in ways that demand our full attention. Today, we're going to go deeper. Our faculty is going to take us through the science, the clinical landscape, and the exciting developments on the horizon. Before we begin, bring in our first presenter, I'd love to hear from you. We have a quick poll on your screen, just one question to help us understand where you're starting from today. The question is, what brings you here today? Do you want to learn more about who's in the room with us? Your options are, I am a healthcare professional, I am a researcher, I am living with Sjögren's or autoimmune disease, I am a caregiver, or other. All right, thank you. That's really valuable context. Before we go any further, I want to introduce you to someone who's supposed to be with us today, but unfortunately had a last-minute emergency pull her away. Dr. Abigail Koppes, Abby, was our first planned presenter. She is an associate professor of chemical engineering at Northeastern University, a tenured scientist, a marathon runner, a mountain biker, a downhill skier. She's been married for 15 years to her high school sweetheart. She's a dog mom, an aunt, a daughter, a friend, and she's someone who, until a few years ago, thought of herself first and foremost as an athlete. She couldn't join us today, but she wanted her story to be part of the conversation, so she shared these slides with us, and I want to walk you through them. In 2020, Abby had what appeared to be mild COVID-19 infection. What followed was anything but mild. Over the next 3 years, she navigated a cascade of symptoms that were each on their own easy to dismiss or misattribute. Profound fatigue, brain fog, dry eyes so severe they registered 0 on the Schirmeyer test, dry mouth and skin, Raynaud's, flushing, rashes, hives, GI distress, migraines, neuropathies, POTS, and the list kept growing. What she was experiencing had a name, actually several names: Sjögren's disease, mast cell activation syndrome, small fiber polyneuropathy, a cluster of conditions that had been present and interacting for years in a body that medicine had been treating one symptom at a time. Ultimately, she had neuro-Sjögren's, not just dry eyes and dry mouth, the nervous system, the whole body, the whole person. And here's the thing that really stays with me about Abby's story. She is a scientist. She had the tools, the vocabulary, the access to medical literature that most patients do not have, and it still took years. It was still, as she puts it, a long road, mainly because these are invisible and dynamic illnesses. You can't always see them. They shift, they overlap, and when clinicians treat peace in isolation, the patient falls through the gaps. With that context, I'd love to dive into the science to see what's coming next. So let's take a quick poll before our next presenter. This one, Which emerging Sjögren's therapeutic target are you most curious to hear about today? Your options are BAFF inhibition, CD40, CD40L pathways, FcRn inhibitors, patient-reported outcomes and monitoring tools, or all of the above. Wonderful. That's a great segue into our next presenter. Our second presenter is Dr. Mohammad Haj-Dezfulian. Dr. Dezfulian is an assistant professor of pathology and a member of the Institute of Immunology at the University of Pennsylvania. His research sits at the intersection of immunology and technology development with a focus on T-cell biology and the immune drivers of autoimmune disease. Work that is directly shaping how we think about the next generation of Sjögren's treatments. Mo, thank you for being here.
Well, thank you so much, Laura, for that introduction. Can you guys hear me? Perfect. Well, before I start, I'd just like to thank Laura and the organizers, NRF, and others to kind of for inviting me and giving me this opportunity to present our work. So for today's talk, I'm the way I've designed it is kind of in two sections. For the first part of the talk, I'll be telling you about these new technologies that we're developing that's allowing us to unbiasedly kind of look at the immune system and what they're recognizing in various autoimmune contexts. And in the second part of the talk, I'll be telling you how we've applied these platforms in Sjögren's disease, especially, what we've understood and where kind of our research is heading in the next couple of years. I'll start with kind of a brief background to kind of orient us. So the body's immune system, especially the adaptive immune system, is kind of composed of B cells and T cells. Now, these cells have evolved to recognize both pathogenic and neoplastic cells in the body. But one of the difficulties that we've had in the field has been our inability to understand what these immune cells are recognizing in an unbiased manner. If you think about it, kind of our inability to be able to systematically and unbiasedly decipher the specificity has kind of significantly hampered our understanding of both, the basic biology of T cells and the function of contribution of T cells to various disease states and pathologies. For example, in the context of autoimmune cell or autoimmunity, what do these immune cells recognize? Now, the reason it's been kind of difficult for us to understand the contribution of T cells specifically in autoimmunity has been because of the high level of diversity that we have at the level of the TCR, which recognizes kind of an antigenic fragment that's being presented on the antigen presenting cells. You also have this diverse array of HLA. Each one of us has multiple different unique HLA alleles. And we have this library of different antigens that are constantly being processed and presented on any given HLA molecules. So the focus of our research for the past several years has been trying to develop tools and technologies that would allow us to unbiasedly decipher the specificity of T cells. Now, to develop these platforms, I'm not going to be going into too much detail, but basically we've developed two kind of complementary technologies. One is for CD8 T cells. Now, T cells are broadly categorized into two groups. On the one hand, you have CD8 T cells, which are cytotoxic. These are the ones that once they recognize their target cells, they typically kill those target cells. And on the other hand, you have the CD4 cells, which are mostly categorized as non-cytotoxic T cells. And their main function is to help the immune system or the B cells especially to develop autoantibodies or antibodies in general. So what we've been trying to do is to kind of develop technologies that would allow us to unbiasedly decipher their specificity. We initially focused on the CD8 T cells, and we developed this technology called T-scan1. And then we focused on CD4 cells, and we developed another complementary technology called T-scan2. Both of these technologies have kind of three main criteria. One is that they're totally unbiased. In other words, they can accommodate and allow us to assess the entire human proteome. So we can figure out what these T cells are recognizing in these different contexts. The other thing is that these platforms incorporate both endogenous T cell signaling and endogenous antigen processing and presentation. And that's really important if you're trying to understand what specific epitopes are physiologically relevant and they're being recognized by the immune system. Um, so I won't well, just to kind of give you a little bit of a background as to how we've developed these platforms. Now, I mentioned CD8 T cells are cytotoxic. So the reason that they're cytotoxic is once they recognize their target cells, they get activated and they secrete this kind of protease within the target cells called granzyme B, which ultimately results in cell death in the target cell that was recognized by the T cell. To measure kind of how much granzyme B is being targeted or secreted within the target cell, we developed a reporter. This is a fluorescent reporter that's not, we permutated it, so it's not fluorescent. But we've incorporated a granzyme B cleavage site in the backbone of this protein. Once the protein is recognized by kind of this protease, the protease can now cleave this protein. There's no inhibitory signal anymore. So as a result of that, the protein is folding properly. Once the protein is folded properly, you get this red fluorescent signal, which then allows you to figure out which one of the target cell, which antigenic fragment that was present in it was being recognized by the T cell. So what we ultimately do in the context of our screens is that we take our granzyme B reporter, we incorporate it in our target cells. And if you co-culture that with your T cells of interest, either CD8 T cells that are already cytotoxic, or we've engineered CD4 cells that are traditionally not cytotoxic to behave like cytotoxic T cells, once we co-culture them with with the target cells, if the T cell is able to recognize its cognate antigen, the antigen that it's kind of able to is being presented by the APC cell, the T cell becomes activated, and as a result of this activation, secretes granzyme B within the target cell. Once it's within the target cell, then the granzyme B reporter comes on. And now we can kind of use that to sort out the target cells using FACS or flow-based experiments. In the context of a screen, what we're doing now is we're sorting these cells out, the ones that have activated our granzyme B reporter, from all the cells that don't have the granzyme B reporter, and use that for downstream analysis to figure out, what specific antigenic fragment was being expressed or presented by that specific APC cell. A key component of our platform is the antigenic libraries. So in the context of autoimmunity, any one of the 22,000 or so proteins that's being generated by the body could potentially be targeted. And none of the assays that we have right now were able to accommodate this vast antigenic space, or each one of these 22,000 fragments could potentially be antigenic. So the way we've addressed that question is basically we decided to resynthesize the entire human proteome. So in our body, in our genome, we have 22,000 or so annotated proteins. So these are the building blocks of the body. And if you chop them up into small fragments, so if you do this for each one of these 22,000 proteins, if you chop them up in bioinformatically into small fragments, 90 amino acid fragments, you would have 260,000 fragments now. Once we have these 260,000 fragments, again, we can reverse translate these back into DNA. So we would have DNA corresponding to each one of these fragments. These fragments are all overlapping. That would tile across the entire length of the protein. So if you do this for all 22,000 proteins, you have 260,000 fragments that would cover the entirety of the human proteome. Then we can synthesize this as an oligo pool. Basically, this oligo pool would then represent the entirety of the human proteome. And then we can plug it into a lentiviral vector and use that for downstream analysis. In the context of a T-scan experiment for MHC class 1 presentation, that's for the CD8 T cells, these peptides are being presented and processed on HLA class 1 molecules. And for our class 2 antigen presenting cell, we've targeted all these peptides to go into the lysosomal compartment for them to be processed and presented on HLA class 2. So that's kind of another key component of our platform. So on the one hand, you have these reporters that can measure which cells are communicating with each other. And then we've generated the entirety of the human antigenic space. So then we plug it into each one of those cells that has the kind of the granzyme B reporter following the co-culture. If the T cell is able to recognize one of these 260,000 fragments, we can sort those out and use them for downstream analysis. Now, if you look at all autoimmune diseases, if you do GWAS analysis, so GWAS analysis, these are trying to identify what's the main risk allele that's associated with the disease. In almost all of these autoimmune diseases, the strongest risk factor is usually the HLA allele. And what that means is that, most likely a set of antigens or peptides that are being processed or presented on HLA molecules are being recognized by T cells. And this is basically what's driving the pathogenesis in these patients. So what we've been trying to do is to kind of systematically probe this question of what those antigens are in these various disease contexts. One of these diseases is obviously Sjögren's, but it's the same concept across pretty much celiac disease, RA, lupus, diabetes, MS. We're kind of doing this across multiple different indications. If you know what those antigens are that are driving the disease, it's not only important that will help you understand the etiology of the disease, but it will also help you develop more personalized therapies for each one of these patients, or basically understand what cell types are important to drive the pathogenesis in different contexts and different diseases. So for the purpose of this talk, I'm going to focus on our Sjögren finding. Now, in Sjögren's disease, as you guys all know, it's a systemic autoimmune disease. That means it affects multiple different organs. But one of the main clinical manifestations in patients is T cell infiltration or immune cell infiltration within the salivary gland. Now, within these glands, you have these T cells, especially CD4 cells, that are being infiltrated within these tissues, especially at the early stages of the disease development. Now, these T cells are also clonally expanded. A clonal expansion means that most likely they have seen an antigen. And in the context of autoimmunity, that means most likely they've seen some kind of a self-antigen. And based on the work that's been done by multiple groups, we know that there's a direct correlation between the number of T cells, especially the clonally expanded T cells that we see the patients, and how the severity of the phenotypes that the patients kind of exhibit. What's also interesting about this disease is that traditionally, the only cells that present antigens to CD4 cells is your APC, is your professional APC cells. But within the salivary gland of these patients, the ductal cells also have high levels of MHC class 2 or actually class 2 expression on them. So that means, there's a probability that these cells are able to directly present antigens to the CD4 cells. And surprisingly, most of the CD4 cells typically congregate around these ductal cells that have class 2 expression. So everything that we know about the disease seems to implicate the CD4 cells in the pathogenesis of the disease, especially at the early onset of the disease development. So the question that we were trying to answer now is like, we have the technologies, we know T cells are important. The HLA or the GWAS analysis is also pointing to the fact that these T cells are probably the main drivers or the main contributors to the disease development and the pathogenesis in these patients. Can we identify or figure out what these T cells are able to recognize? So that's kind of the central question that we were trying to address. Are any of these T cells, especially these clonally expanded CD4 cells, recognizing self-antigens? And can we use our TScan2 platform to identify those? Now, to answer that question, basically our experimental design was that we extracted memory CD4 cells from the salivary gland of multiple different patients. We sequenced these T cells, so we knew exactly what the TCRs were in each one of these T cells. And we would then synthesize those TCRs and then plug them back into our TScan platform and then screen them against our human peptidome library. So this is the library that covers the entirety of the human proteome. If these T cells are able to recognize self-antigen, they should be able to recognize one of these 260,000 fragments. So we did these experiments in a pooled manner. So we had multiple different T cells and we were screening them against this library of antigenic fragments that we had. And sure enough, from multiple of these T cells, we were able to identify self-antigens. So one pool of these T cells was recognizing this MAP kinase protein. Another pool was recognizing a DNA damage protein. As far as we know, these are the first set of self-antigens that have been described for these CD4, clonally expanded CD4 T cells in Sjögren's patients. So that answers the first question. T cells within the gland are indeed recognizing self-antigens. And more recently, the research group at OMRF has also published and they've shown that some of these T cells are also able to recognize the SSA, SSB self-antigens that patients typically have autoantibodies for. So collectively, kind of so far, we know that these T cells are able to recognize self-antigen. What was interesting is kind of, we were trying to now understand, okay, we know what the self-antigen is that's being recognized by some of these T cells. We also have the single cell sequencing. So basically, we were extracting the glands from these patients and we were doing single cell sequencing and also spatial profiling. And what we were hoping then is to use the antigens that we've discovered to figure out which specific cell type was being targeted by these T cells. Right? I mentioned that we think it was the ductal cells because the ductal cells had class 2 expression on them or HLA-DR expression on them. To answer that question, once we did the single cell sequencing, so this plot over here is basically showing the different cell types that we were able to characterize in these glands. So this is work that was done in collaboration with Blake Warner's group at NIH. You can see there's multiple different cell types. Some of those are the immune cells. Obviously, you have immune cell infiltration, and then you have these epithelial cells, acinar cells, etc. But what I want you guys to focus on is mostly these darker blue cells, which are our ductal cells. Now, if you look at the ductal cells, I'm not sure why the plots are looking like this here. The cells are not showing up. But the ductal cells have class 2 expression on them, mostly in the Sjögren patients. Sorry about that. There's some kind of a formatting issue here. I think it's a PC and Mac issue. But now if you look at them basically in Sjögren patients, over here you had more dots, which meant that there is more class 2 expression on them. What was interesting is that most of the antigens that we discovered, both the MAP kinase and the DNA damage protein, were also expressed within these ductal cells, right? So not only we were able to identify the antigen, but we were able to show that the antigens were also expressed mostly within these ductal cells. Now, at this point, we can't rule out the possibility of that these antigens are being processed and presented by the APC cells that have infiltrated into the tissue, but we think the ductal cells also have the capability to process and present antigens directly to the CD4 cells. And that explains why the CD4 cells are honing into this tissue and kind of congregating around these ductal cells. Now, the other question that became really interesting, it was like, okay, when I showed you the GWAS analysis, in almost all of these autoimmune diseases, there's usually one HLA allele that's strongly associated with the disease, right? What that means is that most likely that HLA allele is presenting an antigen to the immune system. Now, that's the case for Sjögren and lupus. But the antigens that we were discovering, for example, the MAP kinase and the DNA damage proteins, they had different HLA allele restrictions. And that was kind of a little bit puzzling for us. Is like, okay, well, if the T cells are recognizing, and if the HLA-DR3, that's the main risk allele, is the main one driving the disease, the antigens should be mostly confined to HLA-DR3. Why are we finding antigens on multiple different patient-specific HLA allele? And that's the kind of where things got a little bit more interesting. So, one possibility is that the T cells that have infiltrated and we're studying, they are the secondary T cells. These are not your primary T cells, the ones that recognize especially antigens on kind of on HLA alleles other than HLA-DR3. But it's possible that there is something more fundamental happening, in this case that we don't know. So we went back to the original GWAS analysis that had been done in these patients. So the GWAS analysis, like over here, had shown that this kind of the strong risk factor was HLA-DR3, the HLA allele. Now, what's interesting is within the HLA region, within this region on the chromosome 6, that has HLA, right adjacent to it, you have complement genes. Okay. And these GWAS analyses are mostly done in a European population background. In the European population background, the C4 that's associated with the disease, the complement gene C4, one of them is called C4. You have a fair amount of copy number variations within the C4 genes. Now, the C4, so that means there are individuals that have multiple copies of C4, and there are individuals that have one copy of C4, or sometimes they have an endogenous retrovirus inserted. So they have non-functional complement genes. Uh, in most of the patients, almost 80% of the patients that have the DRB3 allele in the European population background, they also have a C4a null allele. So there's basically two genetic components that could potentially be contributing to disease. It's not just the HLA-DR3. So the question becomes, now, is the signature that we're seeing here, is the association due to HLA allele, or is it due to the complement genes that's within that kind of chromosomal location? Now, if you go back, so this is something that was done in Steve McCarroll's group, and Nolan had done this, Nolan Kamitaki. Basically, what they did to answer this question was that they went outside of the European population and they repeated those experiments. Now, if you go and do these analysis in an African population background, you have a low linkage disequilibrium between the C4 and the HLA allele. Low linkage disequilibrium basically means that these two things are inherited separately. And surprisingly, most of the phenotypes that now we see are now associated with C4 and no longer with the HLA allele. So that means most likely what's driving the disease in these patients is low levels of complement and not necessarily the HLA allele association. Okay. But then the question becomes, why do we have T-cell infiltration? And does that explain why we are seeing multiple different antigens being presented on multiple different HLA alleles? And I think that's kind of in line with what we're seeing in general, because if you have low levels of complement, now complement is expressed, and one of the main functions in the body, it's part of the innate immune system, and that is to clear cell debris from cells that are dying, and also pathogenic fragments. So if you have low levels of complement, you get accumulation of antigenic fragments on multiple different HLA, and they could be presented in multiple different HLA alleles. As a result of that, most likely these antigens are going to be recognized by different T cells on different HLA alleles. So ultimately, and I think that's exactly what we're seeing. So I think our model basically backs this, or our data is backing up this model, whereby the complement is most likely initiating, or the main pathogenic driver in these patients. And then you have the T cells coming in secondarily, coming and recognizing the self-antigen. And it's the interplay of both of these that's driving the development of Sjögren. And that's the same phenotype that we see also in lupus patients, both systemic autoimmune diseases. Now, something else that we kind of did as well is like, okay, well, we were able to figure out that these T cells are recognizing self-antigen. We were able to figure out that which cell type that they're being presented these antigens on. And also we were able to show that most likely complement is driving. But one of the key questions was ultimately, what's the source of these T cells? Where do these T cells come from? Could some of these T cells also be recognizing self-antigen? One of the models that's always been proposed in autoimmunity is that you have T cells that are potentially cross-reactive. Cross-reactivity means that there's a T cell that can recognize multiple different antigens. Now, if these antigens, one of them is bacteria and one of them is self, That cross-reactivity could be what actually ultimately breaks tolerance, results in the breakdown of tolerance, and the ability of that T cell to recognize the self-antigen and the development of autoimmunity. To figure out, to address that question, what we did is we developed this other kind of technology whereby we could take the epitopes that we had identified, for example, the MAP kinase, we would mutagenize these. We would basically make a lot of random variants of these epitopes, and then we would also screen the TCRs against this. And what that allows us to do is basically generate a footprint of everything that the TCR is able to recognize. We know it recognizes epitope 1, but we have all these hundreds of different variants of the same epitope. Could that then help us determine what other epitopes you could recognize? And sure enough, when we did this experiment, we were able to see that some of the T cells, the ones that were recognizing MAP kinase, seem to be cross-reactive with another group of bacteria. So the TCR that was recognizing MAP kinase was also recognizing this group of epitopes that's conserved across multiple different bacterial species. Surprisingly, the abundance of these epitopes is higher in the oral microbiome of Sjögren patients relative to healthy controls. Now, at this point, we can't really say, what's actually driving the disease. Is it like, it's a chicken and egg scenario. So the clinical significance of this observation is not clear to us right now, but we're confident that these TCRs are able to be cross-reactive. Just to summarize, basically, we've developed two different technologies, TScan1 and TScan2. These are the first genome scanning platforms that allow you to decipher the specificity of T cells. And we think our data is showing that these T cells are recognizing, first of all, self-antigen. We think the interplay of complement, the low levels of complement, and also CD4 cells is essential for the development of pathogenesis in these patients. With that, I'd just like to thank, my lab, especially Spanta and Izzy, two really talented postdocs that have done a lot of the work that I kind of described here, and also my former lab members, especially Tomasz, which is kind of my partner in crime in developing these technologies. Thank you so much. I'll be happy to answer any questions. I don't know if it's right now or it's kind of later on, but.
Excuse me. Thank you, Mo. I think we're going to do questions in a little bit, but thank you so much. That forward-looking perspective is exactly what this audience needs, and I know it's going to inform some great questions in our Q&A. Please keep those questions coming in the Q&A box. We're almost there. But one more poll before we hear from our final presenter. This one is, which systemic manifestation of Sjögren's do you find most challenging to identify or manage in your practice? Your options are interstitial lung disease, peripheral or central neuropathy, fatigue, renal involvement, vasculitis, or other. All right, that's really helpful. I'll make sure our next presenter has a chance to speak to that. Let's bring her in. Our final presenter is Dr. Rachael Gordon. Dr. Gordon is a physician scientist and assistant professor of medicine at the University of Pittsburgh, where she also directs the UPMC Sjögren Center. Her research focuses on the immunopathology of Sjögren's disease, including IL-12 signaling as a potential treatment target, and she is building one of the country's most comprehensive translational research programs in this space. Dr. Gordon, welcome.
Thank you for the introduction and for the opportunity to speak to all of you today. And I think this will be a fun bridge to Mo's talk, linking the basic and translational immunopathology of Sjögren's with what we're seeing in the clinic. Next, perfect. So we've discussed briefly during this presentation today that Sjögren's is an autoimmune disease where the immune system is overactive and has a predilection for attacking exocrine glands, specifically the tear and saliva producing glands. But the dirty little secret of Sjögren's disease is that Sjögren's can impact nearly every organ. And I think that this is an important point when we think about the diagnosis of Sjögren's disease, but also how we monitor our patients over time. And I want to give you a flavor of what Sjögren's looks like systemically beyond dryness. So pulmonary involvement occurs, and I will say, I'm going to give you some epidemiology here, but the reality is that we still don't know from an epidemiologic perspective the incidence and prevalence of many manifestations in Sjögren's to the degree that we should up to this point, given how common this disease is. So we see ILD probably in about 20% of our patients. We historically think about ILD and Sjögren's, if we open that textbook, as being lymphocyte infiltrative pneumonias, those thin-walled cystic structures that we see in the glands. But when we actually look at the numbers, it's really NSIP pattern that we see most commonly in the context of Sjögren's. So I just want to highlight that point that NSIP is the most common flavor of ILD that you're going to see, and that LIP really isn't, even though it's a little bit more specific. We do see pleuritis and Sjögren's, albeit to a lesser degree, and pulmonary hypertension, especially with concomitant scleroderma. But the other piece I want to highlight from pulmonary manifestations is it's not just the lung parenchyma, but the bronchial tree can be affected. So we do see xerotrachea in the context of Sjögren's, dryness of the bronchial tree, but we can also see a clinical manifestation that very much mimics asthma. We do see airway hyperresponsiveness in Sjögren's. And what these patients look like are the patient that comes into your office and has that horrendous, intractable cough, as one example. Musculoskeletal manifestations of Sjögren's are incredibly common. Over 70% of our patients have arthralgias with an inflammatory flavor. A much smaller percentage of those patients are going to have clinical synovitis, inflammation of the joints that you're going to see in your exam room. If I were a betting person, we put an ultrasound probe on those patients. I suspect that many of our patients that give a good story of inflammatory arthritis but don't have bona fide synovitis on our exam, that those patients would have ultrasound findings. But inflammatory arthralgias can affect nearly 70% of our patients. Many of our patients will endorse myalgias, just generalized muscle pain. What I will say is a much, much, much smaller percentage of those patients will have a bona fide myositis. So I think we just need to make that critical distinction that myositis, inflammatory muscle disease in the context of Sjögren's, we see it, but it's rare. Neurologic manifestations of Sjögren's really run the gamut and come in three different flavors. The first is peripheral neuropathies. We can see involvement of large fibers that can be detected, large fiber neuropathies that can be detected on EMG, but we can also see involvement of small fibers resulting in small fiber neuropathies, which are far more common in many ways, but won't be detected on EMG. And that's particularly frustrating to diagnose. And to diagnose these, we need to use pseudomotor testing, QSART testing to do that, or skin biopsy, where a pathologist will look for cutaneous nerve fibers or the lack thereof to make a definitive diagnosis. Neuropathies in Sjögren's are incredibly challenging to treat. And we don't have fantastic studies on therapeutic agents, both in large and small fiber neuropathies, to guide our treatment decisions, which is frustrating to our patients and to us as clinicians. We can see central nervous system involvement, and generally this will manifest as demyelinating disease, sort of akin to multiple sclerosis or NMO with optic neuritis findings. One major challenge in our Sjögren's patients is cognitive dysfunction. And the term that you'll hear most commonly in your clinics is brain fog. We may want to lump this in with more bona fide CNS involvement, but we really don't understand the pathogenesis of cognitive dysfunction in Sjögren's. And this is an area where we really do need more data, both to characterize what that cognitive dysfunction is and what's actually causing it. From a hematologic perspective, we see a variety of different hematologic manifestations of Sjögren's. Cytopenias run the gamut from bona fide anemia to thrombocytopenia and to leukopenias, with more specifically lymphopenias. Hypergammaglobulinemia is common in an endotype of Sjögren's, a subgroup of Sjögren's patients that are very much B-cell hyperproliferative. Of all of our rheumatic diseases, Sjögren's has the highest risk of transition to a lymphoma, for lymphomatous transformation. To give you a sense of this, about 5% of Sjögren's patients will develop B-cell lymphomas sometime during the course of their disease. Those B-cell lymphomas are often in the salivary glands, but not always. And they tend to be indolent, but not always. And there is some debate in the field as how aggressively we treat these B-cell lymphomas. And our European colleagues would argue that we should be more aggressive in treating these lymphomas than some providers have been up to this point. When we think about risk over the general population, I mentioned 5% will develop lymphomas across their lifespan. This equates to a risk of anywhere from 10 to 20% 10 to 20 fold over the general public, just to kind of give you an idea of relative risk. Renal manifestations of Sjögren's also run the gamut. And I think there's some idiosyncrasies here that we have to be aware of. We do see interstitial nephritis in Sjögren's disease. Many of us don't biopsy kidneys all that often, right? So we are probably underestimating the incidence and prevalence of interstitial nephritis in this patient population. Moreover, we don't know what the clinical sequelae of interstitial nephritis is in Sjögren's, right? In lupus, it's a little bit easier for us to conceptualize, as our patients with bona fide active lupus nephritis will go on to true and sometimes rapid end organ will occur true and sometimes rapid end organ damage. And in Sjögren's, the flavor of the interstitial nephritis is a little bit different in most patients. And we really don't know what that means in the long haul. We also see electrolyte abnormalities in Sjögren's, specifically acidoses. And these generally are a result of tubular acidoses in the context of kidney disease and Sjögren's. And again, this is an area of active exploration where we're not sure if it's the interstitial nephritis that's directly resulting in these RTAs, or if antibodies to different ion channels and channels involved in water handling and acid-base handling in the kidney are actually driving these acidoses. Glomerulonephritis is seen in Sjögren's, but when we see it in Sjögren's, it's generally due to a cryogammaglobulinemic vasculitis. And so this is where it differs from what we see in lupus. And so almost all GNs in Sjögren's are due to a cryovasculitis and should be treated pretty aggressively. And many of those patients, we will also see neurologic involvement due to vasculitic injury from the sequela of vasculitis involving the nerves, as an example. So minor neuritis multiplex would be an example of that. The skin can also be readily involved in Sjögren's, and the list of cutaneous manifestations that are listed here, it just scratches the surface, right? So we do see malar and photosensitive rashes in Sjögren's, and just because you have a malar rash, we should not label you as having lupus, right? We have to be a little bit more discriminate than that. We can see cutaneous vasculitis, leukocytoclastic vasculitis type rashes in Sjögren's disease, and we see it rather often. And those are the patients where I become, where I have my spidey sense in the background to think about both neurologic and renal sequela as well. Those again tend to be the B-cell hyperproliferative patients. We do see Raynaud's phenomenon, and Raynaud's phenomenon in the context of Sjögren's can precede onset of sicca that we should just be mindful of. And I want to highlight that Sjögren's is really a disease that impacts predominantly women, and predominantly women across their lifespan. So anti-SSA antibodies, or Rho antibodies, that prototypical autoantibody or immune marker that we see in Sjögren's, can cross the placenta during pregnancy. And these antibodies can drive fetal heart block in the fetus, and they can drive rashes, which we call neonatal lupus, in the infant once the infant is born. And the use of hydroxychloroquine, as an example, reduces the incidence of congenital heart block in these patients by 50%. So it's important that if we have younger Sjögren's patients that have the prototypical autoantibodies associated with Sjögren's, that we provide clear counseling and intervention to this patient population. And that intervention is not only hydroxychloroquine, but fetal heart monitoring later in pregnancy. So where are the diagnostic gaps in Sjögren's, and what's the driver of these diagnostic gaps? So a major issue in Sjögren's is that diagnosis is delayed. And there are a variety of reasons for this. The first is symptoms are treated in isolation, right? You go to your eye doctor for dry eyes. You mention your dry mouth to your dentist. If you have some numbness and tingling, maybe you talk to your neurologist. If you're short of breath, maybe you see your pulmonologist and get a diagnosis of COPD, right? But because care is so fragmented in that way, it takes somebody with a background of what Sjögren's looks like to start putting the pieces of the puzzle together to make a clear diagnosis. Many of the symptoms of Sjögren's are non-specific. So if I were to ask the audience for a poll that we don't have set up, how many of you are tired, are dry, have dry mouth? My mouth is dry from talking for the past 20 minutes, and have a little bit of joint pain, right? I would argue that over half of you would raise your hand. And this is an issue because, probably, Sjögren's may be enriched a little bit more in this group than others, but most of you don't have Sjögren's. And so when you have these symptoms that are not specific, it becomes increasingly more challenging. The other piece is that in order for you to have the sensation of dryness, 50% of your glandular function's already gone, right? This is it. The sensation of dryness is a later manifestation. And if you've been dry for a long time, many times you're not even aware that you're dry. So I have patients that have severe dry eye disease with corneal abrasions, and they don't tell me their eyes are dry. I can ask them 10 different ways, and my eyes are fine. But you have them see an ophthalmologist, and they already have ocular surface damage. The other piece is that while dryness is a hallmark feature of Sjögren's, as we've discussed today, Sjögren's is so much more. And in some cases, other manifestations of the disease will occur before a patient feels dry or has objective dryness. And so I'll give you an example in the rheumatoid arthritis world, and then I'll translate that to the Sjögren's world. So for those of you who are rheumatologists in the call, this doesn't happen often, but you'll sometimes have a patient with interstitial lung disease that when we work them up and we also take a proper history, we figure out that they have rheumatoid arthritis. And their ILD, their interstitial lung disease, happens to be the first clinical manifestation that's detected. And we see something similar in Sjögren's. So a consult that I get pretty regularly from from the ILD team at UPMC, so we have a patient with new interstitial lung disease, NSIP pattern. They're reporting some dryness. We've sent off serologies. We're waiting for them. We're waiting for them to result. But I'm worried that this patient has Sjögren's. Can you do a once over? And if the patient has Sjögren's, then that's really an impetus for us to treat them with immunosuppression to prevent progression of their ILD. And so making that diagnosis is important. The other clinical manifestation where this is particularly prominent, where we see a systemic manifestation before dryness, is in our patients with neurologic Sjögren's. The other piece is that serologic testing in Sjögren's is not perfect. So anti-SSA or Rho antibodies are positive in about 70 to 80% of Sjögren's patients. But we get a lot of that data from registries, which need to use some criteria to enroll patients. And when your diagnostic tools are limited and you're reliant on serologies, that's obviously going to enrich for a serologic population. So anti-SSA and Rho antibodies are not the be-all, end-all. If you have a high index of suspicion for Sjögren's in a patient that doesn't have prototypical autoantibodies, it's important to continue that workup with salivary gland ultrasound and a definitive lip biopsy. And so this has been a barrier because if patients are seronegative, completing the rest of their diagnostic testing is actually pretty challenging. And then both in rheumatology and external to rheumatology, there's still an educational gap in Sjögren's. So historically, even within rheumatology, Sjögren's has been thought of as a disease of dry eyes and dry mouth, where morbidity is there, but the mortality issue really isn't impacted to the degree that we see in lupus and scleroderma and inflammatory myopathies with associated lung involvement as an example. So we'll just treat you symptomatically and, that's it, right? And we don't have any immunosuppressive medications that have been shown to address Sjögren, so I don't have much to offer you. So rheumatologists even, and this is a culture that is shifting and changing, historically have not put as much attention into Sjögren's as they have other rheumatologic diseases. And then outside of rheumatology, even many PCPs and ophthalmologists have only heard about Sjögren's in passing and don't recognize its prevalence. And so it's on us as a community to provide this level of education within rheumatology and beyond so that we share what Sjögren's is and the fact that it's a serious and systemic rheumatologic disease. I think we've already talked about the cognitive symptoms of Sjögren's, and that brain fog is a prominent clinical manifestation, one that is hard to treat, and again, not specific. And so many patients that have significant brain fog and fatigue are dismissed by people in our profession, and we have to be aware of this. So if you're a primary care doctor here, or an ophthalmologist, or even a rheumatologist that doesn't see a lot of Sjögren's, what are some of the red flags to watch out for that may make you think about Sjögren's and to pursue a more defined workup? So unexplained fatigue and sicca symptoms in middle-aged women is something to be mindful of. And it's helpful if we can define not only sicca symptoms, but objective dryness in those people. Recurrent unexplained parotid swelling, unilateral or bilateral. Peripheral neuropathies without a clear cause, especially when they co-occur with sicca symptoms. One thing I will point out, though, is that the glands are highly innervated. So even patients with peripheral neuropathies can have dryness and not have Sjögren's. So I just want to highlight that because that's where this becomes quite challenging to think about and to diagnose. Unexplained ILD in a non-smoker, right? I gave you a couple of examples of that already. Positive anti-SSA antibodies in patients that are not dry, right? In our diseases, autoantibodies precede clinical manifestations on the order of years, right? We've had fantastic data supporting this idea. And so those are patients that you're going to want to monitor in an objective way. And again, renal tubular acidoses and stones without a clear underlying etiology. And I actually get quite a few of these referrals from nephrology as well, especially when the RTAs are recalcitrant. And occasionally we do diagnose Sjögren's. The other piece I want to highlight is that Sjögren's comes in two flavors. The first is what we've historically called primary Sjögren's. Sjögren's that occurs without another systemic autoimmune disease. If the patient has single organ autoimmune disease like thyroiditis or primary biliary cholangitis, we still call it primary Sjögren's. We also have Sjögren's that occurs in the context of another systemic rheumatologic process. The most prototypical are rheumatoid arthritis, lupus, and systemic sclerosis. We historically have called this secondary Sjögren's, but we've really changed this nomenclature because we don't use this nomenclature when we have overlap diseases in any other area of rheumatology. So we now call it Sjögren's lupus overlap or Sjögren's rheumatoid arthritis overlap. And this is something that we need to be mindful of, is to make sure that we're not missing another clinical overlap entity when we evaluate these patients. The other piece is that we need to be cognizant that many diseases mimic Sjögren's, right? We talked about many of the clinical manifestations being nonspecific. And I want to highlight that we need to make sure that our workups are thorough to rule out mimics. Okay. So I want to give you a flavor of how we approach Sjögren's treatment in the next 2 or 3 minutes together. I know that we're abutting time. And so the point I want to drive at here is that we need to treat dryness in Sjögren's patients aggressively. And we're targeting symptoms when we do this. And this could be the use of topical agents for dry eyes, mechanical stimulants to promote saliva production, saliva substitute, and as second line, using secretagogues, medications like pilocarpine and cevimeline, to name a few. When we see involvement of other organs, that's really the impetus for us to escalate therapy to a conventional DMARD or a biologic. And up to this point, because we do not have any FDA-approved disease-modifying therapies for Sjögren's, we need to borrow from other diseases in rheumatology. So as an example, if a patient has ILD, we may use a little bit of Cellcept that has shown efficacy in ILD in the context of systemic sclerosis and myositis and rheumatoid arthritis, and second line using rituximab, just to sort of name an example in a framework. So we take the clinical manifestation, we find what other rheumatologic has a similar manifestation and has known therapeutics that impact that manifestation, and we adopt them for Sjögren's. My hope is that this landscape is going to change in the next 6 months when we're likely to have our first disease-modifying therapies approved for the treatment of Sjögren's by the FDA. And then lastly, care needs to be multidisciplinary. We can't just have a rheumatologist running the show or a dentist running the show or an ophthalmologist running the show. A rheumatologist, a dentist, and an ophthalmologist is the bare minimum of a multidisciplinary team. And then based on the clinical manifestations of Sjögren's, you may need help of our pulmonary colleagues, our neurology colleagues, or our renal colleagues. And care should be coordinated, and the team needs to have the right group of experts to address that patient's clinical presentation. I've just mentioned to you sort of the framework of the treatment guidelines of our European colleagues, our EULAR colleagues. The British Rheumatology Society has also put together treatment guidelines that are quite similar and sort of use the same framework. What I'll also mention is, while extraordinarily rare, we do see Sjögren's in kids as well. And what Sjögren's looks like in kids is very different. And the British Rheumatology Society was one of the first groups to actually provide some recommendations based on expert consensus on how to address Sjögren's in the pediatric population. So it's important that we have the right team in place to take care of our Sjögren's patients, and we monitor them not only for progression of their dryness, which I will argue is not benign, but for the other organ systems that can be impacted by Sjögren's, and make sure we catch those systemic manifestations early and we treat them aggressively. And we now have tools that we use predominantly in clinical trials, but I see a role for them in clinical practice where we can use, as an example, the SDI tool for disease activity is a tool that we can adopt, at least in part, to at least make sure we're addressing every relevant organ system in the context of Sjögren's. We can use the ESPRI questionnaire, which is a patient-reported outcome that asks patients about pain, dryness, and fatigue, to get a sense of how our patients are feeling and how our interventions are impacting how they're feeling. And again, making sure that we monitor serologic characteristics of Sjögren's and make sure that we appropriately, and at least on a yearly basis, think about lymphoma surveillance. And with that, I think we can open up the Q&A.
Okay, all right. Thank you, Dr. Gordon, so much. The guideline context and clinical red flags are particularly good things to know about, and I'm sure people were taking notes on them. We're so glad to have these insights from our experts here today, and we've run out of time to answer your questions live, unfortunately. We'll address these on an upcoming episode of our podcast in the room and make sure that you all have the link. As we bring today's session to a close, I want to say a sincere and heartfelt thank you to our presenters. I also want to thank our supporters at Johnson & Johnson Innovative Medicine for making today's independent educational program possible. A few things before we let you go. This webinar will be on demand. We'll send you the recording link and a follow-up email. Please feel free to share it with colleagues who weren't able to join us today. And we have more incredible webinars coming up. The Bigger Picture, CAR T Therapy and Future of Autoimmune Care on Wednesday, May 28th. We hope you'll join us. Registration details will be in your follow-up email and at curearthritis.org. Finally, and importantly, if you are claiming CME credit for today's live session, here's what you need to do. Please scan the QR code on your screen now to access information on the credit claim portal. Once you're there, enter the code shown on the screen to confirm your attendance. We'll send these instructions by email as well. Thank you all so much for being here today and for the incredible work you do for patients living with autoimmune diseases. Please follow us on social media to stay connected, and we look forward to seeing you on May 28th. Take good care and goodbye for now.